Today we will discuss bacterial chemotaxis. Chemotaxis is the directed motion of an organism toward environmental conditions it deems attractive and or away from surroundings it finds repellent. Bacterial chemotaxis is the biasing of movement toward environments that contain higher concentration of beneficial chemicals or lower concentration of toxic chemicals. The signaling pathway that is involved has long been viewed as a paradigm of histidine aspartate-phosphorelay signaling, and it is one of the most well understood physiological processes in biology. The pathway is composed of chemoreceptors, the histidine protein kinase, chemotaxis protein A, and two diffusable response regulators, chemotaxis Y and chemotaxis B. Chemotaxis Y controls flagellar motor switching, whereas chemotaxis B controls chemoreceptor adaptations. Chemotaxis is thought to be involved in pathogenicity, symbiosis, biofilm formation, and stability, and in maintaining bacteria in their optimal environmental niche. The chemoreceptors and other proteins of chemotaxis signaling pathway localize to specific region of the cell as large higher order arrays. Many bacterial species have several chemosensory pathways as well as further components that might be expressed under particular environmental conditions to allow bacteria to tune their responses to a specific environment. The correct interplay between chemotaxis and other sensing system is essential for bacterial survival in a changing environment. Before starting with the signal transduction pathways of chemotaxis, it's very important to understand the modes of swimming behavior in bacteria. Bacteria such as Escherichia coli and Salmonella typhimurium have two main swimming patterns. One is smooth swimming in a rather straight line, a run, and the second is a brief but abrupt turning motion, a tumble. So, two main swimming patterns are a run and a tumble. As you can see here, this is tumbling mode of swimming and this is running mode of swimming. So, a run is the consequence of counterclockwise rotation of flagella. Because of flagellar left-handed helicity, counterclockwise rotation exerts a pushing force on the cell. Since flagella around the cell have different lengths and their distribution is also not symmetric, the net force is not zero. So cell moves in the direction of the net force, right? Then a tumble. The tumble is the consequence of clockwise rotation of the flagella. Here, in the case of tumbling mode. Unlike counterclockwise rotation, which stabilize the left-handed form of flagella, clockwise rotation destabilizes the left-handed helix. So the flagella undergoes a transition from left-handed helix to a right-handed one. So this transition propagates from the flagellar junction with the cell body towards the distal end of the filament. In the absence of stimuli, the tumbles usually occur once every 1 to 5 seconds, depending on the bacterial strain. Next is signal transduction pathway of chemotaxis. So we'll discuss signal transduction pathways in bacterium Escherichia coli. Signal transduction pathways starts with the receptors. When a receptor receives a signal, so location of the chemotaxis receptors and the flagella are different. For example, here in the case of E. coli, you can see here, the receptors are clustered at the bacterial poles, right? Whereas the flagella are randomly distributed around the cells. This prevents direct interaction between the receptors and the flagella, and the communication between them is carried out by a sophisticated signal transduction system, which belongs to large family of two-component regulatory system. So first of all, we'll start with the components of signal transduction pathway. The components of conventional signal transduction pathways are chemotaxis receptors, and the switch proteins. In addition, there are components that may be required for signal transduction, but it's not yet known how. There are two kinds of this type of receptors. One is chemotaxis specific receptors, and second is dual function receptors. Another important thing, that is genes that controlling chemotaxis. So I would like to tell you about these genes that controlling chemotaxis. The genes involved in controlling chemotaxis are AER, chemotaxis A, chemotaxis B, chemotaxis R, chemotaxis W, then Y, Z, then FLIG, MN, TAP, TAR, TSR, and TRG. These genes form a master operand whose gene products control the expression of genes involved in flagellar synthesis, motility, and chemotaxis. This master operand is itself positively regulated by the intracellular levels of CAMP, means cyclic adenosine monophosphate, and its receptor. In this manner, the metabolic state of the cell is linked to the expression of the motility and chemotaxis components. So first is chemotaxis specific receptors, right? Chemotaxis specific receptors, dipeptides termed MCPs, means methyl accepting chemotaxis proteins, are expressed by genes. And these genes are AER, TAP, TAR, TSR, and TRG genes. The methyl accepting chemotaxis proteins are closely related to each other both in amino acid sequence and in structure, right? They are, however, different with respect to their abundance and the sequence of their periplasmic part and the presence of the binding site for chemotaxis R in their cytoplasmic part. Now, what is this chemotaxis R? It's a specific methyl transferase having role in adaptation, right? Chemotaxis R binding site is a specific pentapeptide at MCP's C terminus. Thus, most abundant MCPs at TSR and TAR possess this chemotaxis R binding site and therefore can function independently of the other MCPs. The other MCPs which do not possess this binding site depend on the presence of TSR and TAR to function in adaptation. Now listen, therefore, to be functional in adaptation, the minor MCPs must interact with major MCPs, and this may be one of the reasons for the organization of the receptors in cluster. Next is dual function receptors. Chemoattractant sugars do not bind to MCPs directly. They either bind to specific periplasmic binding protein involved in both chemotaxis and transport of sugars, for example, galactose, maltose, and ribose binding proteins, or they bind to specific enzyme too, for glucose, mannose, mannitol, and others of the phosphoenol pyruvate dependent sugar phosphotransferase system, that is PTS. The responses to both types of sugar chemoattractant are, however, mediated by MCPs. The periplasmic binding proteins bind to specific MCPs, specific MCP like TAR or TRG, and thus elicit the chemotactic signal. In the case of PTS sugar, the PTS enzyme 1 modulate the kinase activity of the complexes MCP chemotaxis W chemotaxis A. Like MCPs, at least some of the dual functional receptors like periplasmic maltose binding proteins are clustered at bacterial poles properly in order to allow direct interaction with the MCPs. Next is switch proteins. The switch, a complex of three proteins, FLIG, M, and N, is the target of signal from the receptors. It is known that each protein in the complex interacts with other two proteins. The complex is linked to MS ring of the flagellar motor via FLIG, forming the switch motor complex. Next, I will tell you about the proteins mediating receptor that switch communication within the cell. The molecule that delivers the clockwise signal to the switch motor complex is chemotaxis protein, chemotaxis Y. This protein is the response regulator of two-component regulatory system and can be in two main states, phosphorylated and non-phosphorylated. The phosphorylated site is ASP57. Chemotaxis Y is phosphorylated by chemotaxis A, an autophosphorylatable histidine kinase. The autophosphorylation site on chemotaxis A is HIS48, and it is only present in the long form of chemotaxis A and chemotaxis AL. Phosphorylated uh, chemotaxis Y is dephosphorylated spontaneously or in enhanced manner by specific phosphatase uh, chemotaxis Z. So the short form of this chemotaxis A and chemotaxis AS forms complexes with chemotaxis Y and chemotaxis Z and activate the later, at least in vitro in lab conditions. Chemotaxis Y has also been phosphorylated by small phosphodonor that reside in the cell, but their contribution is negligible relative to that of chemotaxis A. Next, we'll start with the signal transduction. We'll discuss signal transduction in response to negative stimulation and signal transduction in response to positive stimulation. Under non-stimulated conditions, the phosphorylation level of chemotaxis Y is relatively low, right? Level of this is relatively low. The extent of chemotaxis YP binding to the switch and the probability of clockwise rotation are low. So this situation results in predominant counterclockwise rotation and occasional clockwise rotation. A negative stimulus then shifts the receptor to an active form that together with chemotaxis W, these two, stimulates the autophosphorylation of chemotaxis A. When chemotaxis A autophosphorylate, it intensely phosphorylate chemotaxis Y, which in turn dissociate from quaternary complex. Chemotaxis YP, this has a relatively high affinity for this switch protein FLIM and the phosphatases chemotaxis Z. So binding to the switch results in increased probability of clockwise rotation and therefore the cell tumbles frequently. Binding to chemotaxis Z sequentially results in chemotaxis Z oligomerization and delayed activation of its phosphatase activity. Consequently, chemotaxis YP is dephosphorylated after a delay and the clockwise signal is terminated. Next is signal transduction in response to positive stimulation. Positive stimulation shifts the receptor to a form that together with chemotaxis W inhibits the autophosphorylation of chemotaxis A. Therefore, the steady state level of chemotaxis YP drops down and the probability of clockwise rotation decreases. The outcome of this situation is prolonged runs and rare tumbles. Under certain conditions, chemotaxis Z might be involved in lowering the chemotaxis YP level. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.